In this video, I will be discussing the function of the integrin in the extracellular matrix, and we will be talking also on cell adhesion onto surfaces. So if you can remember, in the previous video, we've talked about the four components of the extracellular matrix, which are the collagen, we have the proteoglycans, we have the laminins, and then we have the fibronectins. Those four are actually found outside of the cell. Thus, they are really part of the extracellular matrix. This time, the integrin that we will be talking about is actually embedded in the membrane of the cell, as you see here. So in a way, the integrin is really attached onto the cell permanently, and it helps the cell attach and interact with the rest of the molecules in the extracellular matrix. So they are transmembrane proteins, and they connect the cell to the ECM. So in a way, their name actually means that they integrate the intracellular and extracellular environments of the cell. So integrins are actually made of two subunits. We have the alpha and the beta. And when they are inactive, they are in this particular conformation. You can see that their upper portion is sort of bent down. Okay, so they're inactive. When they're active, this bending position is actually going to be straightened, and they will become taller, and they can become active based on signals, whether it's from the outside of the cell, from the ECM, so we have an outside-in signaling, or an inside-out signaling, which the trigger of which is from the inside of the cell. So then they become active and they can attach to other parts of the extracellular matrix and connect the cell to the ECM. So when the signal is also removed, the integrins can also go back to their inactive state. There are many types of integrins depending on what type of alpha or beta subunits there are present. So depending on the combination, so as we see here. So different types of integrins have different types of signals or ligands, as also listed here. So we have those that are RGD recognizing, okay, so as shown in this column. So remember in the fibronectin, if you go back to our previous video, the fibronectin has the RGD sequence. This type of integrin that we're talking about here in the first column can recognize that sequence and attach to that fibronectin. Well, there are also integrins that don't have that type of recognition. They cannot recognize the RGD sequence. Instead, they attach to other molecules like collagen without the need for an RGD sequence. Here we can see how the integrins on the surface of red blood cells actually help in the attachment of one red blood cell to another. Okay, as we see here, as the integrins actually sort of catch and hold onto fibrinogen, as we see here, this squiggly fiber in between the red blood cells. And this is triggered when there is an injury. Okay, there's a signal from the injured site, and it will cause the red blood cells that are present in the vicinity to activate these integrins, and the activated integrins will hold on to the fibrinogen, and the fibrinogen will sort of become a mesh and will catch platelets that are passing by. And the platelets will lodge, and there will be a chemical reaction to start forming a plug so that bleeding may be stopped. Okay, so this is largely thanks to the presence of integrins in the red blood cells. Now, there are certain types of people, or there are certain occasions when we don't want blood clotting to happen. Okay? So for example, when there are high-risk vascular surgeries, we don't want blood clots to form as we do the surgery in the wound because that could flow into the other parts of the circulatory system of the patient and might block certain capillaries and can cause further damage. So in order to prevent that, there is this drug, RioPro, and it's actually in the form of an antibody which can block the integrins in the red blood cells from catching onto the fibrinogen, which they do normally when there is an injury. So blood clotting here will be prevented. So that ends our 5 types of molecules that are present in the ECM. 4 of them are strictly in the ECM, and 1, the integrin, is embedded in the cell membrane, which allows the cell to communicate with the outside world. Now let's talk about cell anchorage. How are cells anchored to their environment? So here we have a series of pictures of, I think this is a mouse fibroblast. If you put it in a Petri dish and leave it there for several hours, we can see that after 30 minutes, the portion that touches the glass actually starts to spread. Okay, as opposed to the end that's not touching the glass. After 60 minutes, the cell has spread itself even more onto the glass. After 2 hours, the shape has dramatically changed. It has become flattened and it has sort of spread itself onto the glass entirely. And after 24 hours, one day, the cell has assumed a certain flattened shape and spread still onto the glass surface. So there is this mechanism actually that allows the cell to do this. And really, this is very important because if you're a cell and you're taken out of your native environment and you're placed onto some random surface, you need to secure yourself so that you won't be rolling about. So you need to hold onto that surface, and that's exactly what this mouse cell is doing. It's trying to hold onto the glass surface. So what could be causing this? There's this colorized picture with certain fluorescent elements of a cell that has sort of held onto a surface, just like our mouse fibroblast shown here on top. Notice how there are certain points in orange that are arranged somewhat in an organized way. These are actually focal adhesions. These are the points where the cell is trying to hold onto the surface. These are the hands that are grasping the surface. And the green lines that you see here connected to those orange points are actually actin fibers. And they are the ones that are providing strength with the holding onto the surface. Okay, so we can see here in this even greater magnification of electron micrograph how the actin filaments are actually connected onto the membrane of the cell from the inside. So this is a picture from the inside, and then here on the outside are those orange points that we see here, the integrins. Okay, so take note, it's the integrins that are holding onto the surface, and they are attached to the actin filaments, which provide traction. Okay, and again, this is what we call as focal adhesion. This is what happens when you put a cell out of its environment. You put it on a surface, focal adhesion with the integrins and the actins will happen. Okay, and this is a detailed look at how the integrins, so we can see here the glass surface. And then the integrins are these red ones. Okay, you can see them in pairs. And they are sort of attached onto the glass, holding onto the glass. Okay, like the tube feet of a starfish, right? And they are attached to these fibrous structures, and among them will be actins. So C-actin is providing the traction that's holding on to the integrins. So again, focal adhesion. And here's an interesting picture of a muscle cell that was placed onto pegs, a surface that's sort of pegged. And look how the cell is holding onto those pegs. You can see how force is being applied here so that it's sort of pulling onto some of those pegs. So focal adhesion really generates force. No? You imagine yourself riding a roller coaster. You are the cell, and the roller coaster is about to do a 360-degree turn vertically. You try to hold onto the railing really hard or really strongly. That's what the cell here is doing, focal adhesion. But not with hands, but with integrins and actin. So focal adhesion can actually be actively activated. As we see here on top, there's no focal adhesion yet. But after some time, the focal adhesion would develop if the cell is placed onto a foreign surface. But what about cells that are in their original places, like the cells on your body right now, the cells on the basal lamina? So we can see here the basal lamina. The cells are holding onto the basal lamina with the help of hemidesmosomes. Okay, so the hemidesmosomes are the ways on which the cells that are in their native location, meaning to say you didn't remove them from the tissue, that's how they hold on to the extracellular matrix in that tissue. While the focal adhesion that we just learned about is when cells are removed from their native environment and they're placed somewhere else that's unnatural, like a surface of a glass. If we're going to look at the structure, we can see that, okay, we actually have intermediate filaments here that sort of connect the hemidesmosome into the cell cytoplasm filaments. And then here we can see the integrin. So here we have the cell membrane, this light blue colored layer. And the integrin is an integral protein, and it's sort of holding onto this cross-shaped molecule. And you guessed it right, that's the laminin. And this is the basement membrane or the basal lamina, which also contains the type 4 collagen, if you can remember that. Okay, so this is a detail of a hemidesmosome.